Hello and welcome to Nikolay's genetics lessons, and today's problem I got from one of my subscribers, and here's the problem. If the length of the DNA is 3.4 multiplied by 10 in the power of 2 angstroms, then what can be the number of amino acids present in a protein coded by this DNA? So here is the double-stranded DNA, and this is linear form, and on one strand we may have, say, C, G, T, A, G, C, T, A, T. Then on the corresponding strand of the DNA we would have G, C, A, T, C, G, A, T, T, A. So this is once again a linear representation of the double-stranded DNA. In nature it never happens in this form. Actually, in nature it represents double helix. So it twists two strands of the DNA twist around each other. You may think that here's one turn, here's another turn, here's a third turn, but actually here is one turn. So one turn when one strand of the DNA would make full circular movement around the joint center. So in order to solve this problem, we don't have to count how many turns DNA, double-stranded DNA would make, but just for your information, one full turn DNA makes about every 10.4, 10.5 DNA bases, and one DNA base is about 3.4 angstroms, and as you see, according to our problem, we have around 100 bases. So 3.4 multiplied by 100. That means we have here is the length of one base, and if we multiply by 100 we are going to get 100 bases. So now we have double-stranded DNA with 100 bases, and now we have to find how many amino acids are present in a protein coded by this DNA. And now you have to know another conversion factor, which is 3 bases would code for one amino acid. We call such bases nucleotides or codons. If we know that we have 100 bases and we divide by 3, because 3 bases would code for one amino acid, we are going to get number of amino acids that this strand of the DNA would code for. And this is going to be 33. 33.3, but we have to round to whole number. So this is going to be our answer today. But I have one more comment for you. Actually, different variants of the answers possible. For example, take a look. Here is the structure of the messenger RNA that code for the protein, and every messenger RNA starts with 5' cap. It doesn't start with this cap. This cap would be added to the messenger RNA after it would be expressed. Then we have a fragment that we call 5' UTR, 5' untranslated region. It's not going to be translated into protein, but here we would find a regulatory sequence. And Here is a fragment that codes for the protein. Here we have start codon, which is AUG. Here we have stop codon. There are 3 variants of the stop codons. So here's one codon. Here's another one with which translated sequence would end. And once again, here we have 3 prime UTR. This is untranslated region that also have regulatory sequences. And then this messenger RNA would be modified, and 3 prime poly A tail would be added. So both 5 prime cap and 3 prime and poly A tail would protect messenger RNA from degradation. And now let's return to our coding sequence. As you see, stop codon we can exclude because it doesn't code for any amino acid. So here's another variant of the answer. If we exclude the last codon, which have to be present because any messenger RNA have to have start codon and stop codon. But stop codon doesn't code for any amino acid. So in this case, we are going to get 32 amino acids. And also, we have to take into account that in many cases, the first amino acid that is coded by sequence AUG, which is start codon, which is also code for amino acid methionine, but does it mean that every single protein would start with methionine? Actually not, because in many cases this methionine would be cut away, and this gives us another variant of the length of this protein that would consist of 31 amino acids. So as you see, 3 variants of the answers are possible. But most likely on your exam, this is going to be the only correct answer that your professor is looking for. But I would recommend you to raise your hand on your exam and ask professor if you have to exclude the first codon, that is start codon, that is in many cases would be cut away, and if you also have to take into account that last codon have to be a stop codon, which doesn't code for any amino acid at all. So most likely, as I said, this would be the correct answer. If I wouldn't have an opportunity to ask such questions, I would choose the first answer. But I show you that actually different variants of the answers are possible. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.